Hi, everyone. I am going to do a recording of cell reproduction notes for you guys. I'd done a couple of video recordings for my chem students, and I decided I wanted to do one for you guys in case anybody was confused reading the Khan Academy stuff, because I know some of you learn better when you can hear an explanation as well. So we're going to go through this notes presentation, and this is talking about DNA, chromosomes, and mitosis. I'll do another one for meiosis in a little bit. So what forms can DNA take? Well, there's a few of them. You may have heard me mention the term chromosomes before, but you probably haven't heard me talk a whole lot about chromatin or chromatids. So chromatin is another word for just regular DNA and the structural proteins that surround it. So if you've ever thought of like an uncoiled mass of DNA, that's chromatin. It's just this like goopy mess of DNA that you can see down in this photo right down here. It's just a gob of DNA. A chromosome, on the other hand, is still DNA, but it's DNA that's tightly coiled up into a shape that you can actually see. So here in this photo down here, you can see the chromosomes individually. So imagine all this chromatin just basically wound itself up and up and up and up and up and up and up until it became a chromosome. And this term chromatid over here, still DNA, but the difference between a chromosome and a chromatid is that a chromatid is a replicated chromosome. So here in this picture down on the right, we have two chromatids. So this one on top here is the original chromosome. And then as soon as that original chromosome got replicated using DNA replication, then we have the replicated chromatid here. And those two are going to be joined at the center at this little spot here. And we refer to that as a centromere. So DNA is usually in the form of chromatin. It's usually unwound. It's usually just a gob of DNA. Because normally, during normal cell functions, the cell needs access to that DNA if the cell wanted to make a protein or some sort of other structure, whatever, it needs access to specific genes. So that DNA has to be uncoiled and in the form of chromatin. When it coils up into a chromosome, that means that that cell is going to replicate itself or make a copy of itself. And when the DNA is in chromatid form, that's after it's replicated itself. So right imminently before the cell divides, you have chromatids. So here is a graphic that kind of shows you what's going on when DNA is going to turn all the way into a chromosome. So here we have the unwound DNA. It's still in its double helix form. But if that winds up and winds up and winds up and wraps around these little yellow balls called histones, and then winds up some more and winds up some more and winds up some more, eventually you get this really super coiled chromosome here. So a chromosome is, in effect, a single DNA molecule that's coiled up, rod-shaped, and made of DNA and those histone proteins. And chromosomes are found in eukaryote cells. And if you recall, eukaryote cells are the more complicated cells. We, humans, plants, animals, have eukaryotic cells. They are not found in bacteria, which are prokaryotic cells. And you only find them in eukaryotic cells during cell division. Histones, as I mentioned, are these little yellow balls. They are proteins that help chromosomes maintain their shape. The reason that they're there is, think about if you've ever had a rubber band and you kind of twirled it and twirled it and twirled it and still it started to coil up on itself, but that shape was very hard to maintain. Like, it kind of twisted all over the place. But what histones do is they provide a structure for the chromosome so it's not wonky and doesn't work right. And they aid in that tight packing, allowing it to be easier to coil into a chromosome as well. What does a chromosome look like during cell reproduction? Well, we talked about the chromatids, so that again is post DNA replication. That's after that chromosome, that piece of DNA has gone through all of the DNA replication. They form because of DNA replication, in fact, and there is that centromere in the middle that holds the two sister chromatids together until they have to separate in cell division. So each species on planet Earth has a different number of chromosomes. We, as humans, have 23 pairs or 46 total chromosomes. And we say 23 pairs because we have 23 different chromosomes, but we have two copies of each one. So for chromosome 1, we have one chromosome 1 that we inherited from our mom and one chromosome 1 that we inherited from our dad, etc., etc., etc. Some of the other chromosome counts, like a pea plant has 14 different chromosomes, a sunflower has 34, a cat has 38, a pufferfish has 42, human has 46 total chromosomes, a dog has 78 total chromosomes. So if you've ever thought that the more complex the organism, the more chromosomes it's going to have, absolutely not. That is not a thing. Look at human versus dog. I don't think anyone would argue that dogs are more complex than humans, and yet they have 78 chromosomes versus we have 46. And then a pufferfish that's a lot smaller than a human, arguably a lot less intelligent than a human, has almost the same amount of chromosomes that we have. So you really can't tell how intelligent or complex or whatever an animal or plant is based on its number of chromosomes. It just has that number of chromosomes. How are they categorized? Well, first of all, there's within the 23 different chromosomes that we have, there are different types of chromosomes. We have autosomes, which are the first 22 pairs, and then we have the sex chromosomes, which is the 23rd pair. There's some other categories that we can use as well. Scientists are going to use the size of the chromosomes to identify them. So you see chromosome 1 up here is the biggest chromosome versus 22 and 21 down here are really tiny. You can use the banding patterns. So all the banding patterns that you would see on a chromosome, and it's easier to see on an actual picture. This is a computer generated image, but each chromosome is going to have gene bands. And so you can use those gene bands to identify the chromosomes, which is helpful because if you look at the differences between chromosome 1 and chromosome 2, they're not really that different in size. So you would have to take a closer look at the gene bands to really tell the difference. And then third, if you couldn't use the gene bands or if you weren't quite sure, you can use that centromere position on each chromosome to tell the difference because that position is very unique to every chromosome as well, where this chromosome would be attached to its replicant, its sister chromatid. Like I was saying before, your sex chromosomes, that's your 23rd pair of chromosomes, that determines the sex of an organism. For us, if you are a female, you are an XX, and if you are a male, you are an XY. So this karyotype right here, that's what this image is called with this all these chromosomes organized. This image represents a male. If it had two X chromosomes, that would be a female. Now, there are other options. Like every human on Earth has not only either had XX or XY, there are certain genetic disorders that you could have an extra X chromosome. You could be missing an X chromosome. You could be missing a Y, have an extra Y, though that one's pretty rare as far as I know. So errors in that sense do happen. Another error that is easy to point out would be something called trisomy 21. And so what that means is that if you have an extra or a third 21st chromosome, that's what trisomy means. And that particular genetic disorder results in Down syndrome. Autosomes, the first 22, as I was saying before, these are coding for everything else. Your sex chromosomes code for a few things, your autosomes code for every other thing. But don't fall into the trap of thinking that the sex chromosomes only code for sexual traits, such as secondary sex characteristics or something like that. They don't. Um, there the gene for color blindness is actually, or the gene that affects color blindness, I should say, one of them is found on the X chromosome. So color blindness is known as a sex-linked disorder because, and males are actually a lot more likely to get it because they only have one copy of the X chromosome. So if that copy of that gene on the X chromosome is defective, there's not another copy of the X chromosome to potentially mitigate that. They only have one shot of having good non-colorblind vision versus females that have two. So if they had one faulty gene, there's another chance for them to have a healthy gene on the corresponding X chromosome. Um, what are homologous chromosomes? So there's going to be a lot of terminology that I'm going to throw at you, and I apologize for that. But a homologous chromosome is basically two copies of each autosome. So every organism that's produced by sexual reproduction, so that applies to humans, cats, dogs, whatever, they all have two copies of each autosome, which you could see very clearly in the karyotype from the last couple of slides. So let's pretend that this is chromosome, or these are chromosome 1 right here. So the red copy, let's say you got that from your mom, the blue copy you got from your dad. These here are homologous chromosomes because they are the same chromosome, you just got one from each parent. Once both of these go through replication before cell division, now you have what's known as sister chromatids. So you have that chromosome that went through replication you got from your mom over here, and now there's an exact copy joined at a centromere. The chromosome you got from your dad, again, went through replication, and now there's two copies of it joined at a centromere. The two copies of each autosome are called homologous, or you can refer to them as homologs. Homologs, it really doesn't matter how you pronounce it. I usually will say homologous chromosomes. Either way, doesn't really matter. They are the same size, the same shape, they carry the same genes for each trait, etc., etc. What I was saying earlier, what is a karyotype? This is a karyotype. This is actually a proper real image. It is not a computer generated image like the other one was, which is actually helpful because you can see the little gene bands in the chromosomes as you look through it. But what it is a pictomicrograph of the chromosomes in a dividing cell arranged in pairs. So if you were thinking that scientists just aimed a tiny little camera into any cell ever and took a picture and all of the genes were in chromosomes, that's not how that works. They have to aim for a cell that is dividing so that all of the DNA is wound up into chromosomes when they're going to take that pictomicrograph. Because otherwise all they would see is this gob of messy snot-like DNA. And they use the banding patterns to match the pairs, as I was saying earlier. And then here's a review of what they're going to use to organize those. But you can see 1 and 2 look very, very similar, but you can use the patterns, the centromere position, all of that to identify what you have. Like this one here, chromosome 4, has this little white dot on the tip of it that chromosome 5 doesn't have. So that would be one way to identify 4 from 5. And going down, you can see the differences in patterns of each one. Something that you should be aware of going forward, what is a diploid number of chromosomes? So the term diploid means two sets, really. So if you are a diploid cell, you have two sets of each chromosome. So like, let's pretend this was chromosome 1. You'd have two of each of chromosome 1, two of chromosome 2, two of chromosome 3, etc. So this is essentially a normal cell. Normal cells in our body are diploid. A haploid cell, on the other hand, only has one version of each chromosome. So if you were to sit and think about it, you would probably be able to guess which cells in our body are haploid, but for the sake of time, I'm just going to tell you sperm and egg cells are haploid because think about it. Their entire purpose is to make a new human. So they can't be diploid because if you had a diploid with all 46 chromosomes joining with another diploid that had all 46 chromosomes, then the offspring would have what, 92 chromosomes? And that's problematic because extra chromosomes is not better. Extra chromosomes causes genetic deficiencies, abnormalities, and it causes a lot of problems. So our haploid cells, our sperm and our egg cells are haploid so that they can make a new human with the proper number of chromosomes. Um, Something to be aware of with diploid cells, they're referred to as 2N. N is the number of chromosomes you receive from each parent. So N for humans would be 23. In a diploid cell, we have 46 chromosomes, as you're seeing here. So 2 times the number of chromosomes you receive from each parent, or 2 times 23, gives us our diploid number of 46. Haploid cells are just referred to as N because they are only the number of cells that you would, or the number of chromosomes that you would receive from each parent because that's their function. Once again, what is a haploid number of chromosomes? Cells that contain one set of each chromosomes. They have half the normal amount or half of that diploid number. Sperm and egg cells are haploid in us, referred to as 1N. So that brings me next to differences between asexual versus sexual reproduction. Asexual reproduction is the production of offspring from one parent. Humans do not do this to produce their offspring. That's not a thing. Um, we produce our offspring through sexual reproduction, or the fusion of sex cells or gametes, one from each parent. However, there is a type of asexual reproduction that goes on in our bodies, and that is referred to as mitosis, because we're not fusing cells all over the place to repair our own wounds or to grow more skin every day or anything like that. So any singular cell reproduction that happens in our body is mitosis or of an asexual nature. But when we produce full-fledged offspring, we are doing sexual reproduction. So there's some different kinds of asexual reproduction. Binary fission is one example, and that's the division of a prokaryotic cell like bacteria. And binary fission is very, very simplistic. All that really happens in binary fission is your chromatin or chromosome or whatever that is going to make a copy of itself, and then the cell divides in half. And that's really it. Copy of DNA, cell divides in half. Mitosis, or the type of asexual reproduction that we do, is much, much more complicated. Mitosis has many different steps in it. And since our cells are more complex than prokaryotes, there's more things that the cell has to worry about when it's replicating everything. Mitosis also is going to occur in unicellular organisms like amoebas, or it can also occur in organisms undergoing growth, development, or repair. So like I said, if you cut yourself and your body's growing scar tissue, that's happening through mitosis. If one day when you guys have babies, as your babies grow, that's going to be happening through mitosis after the initial joining of sperm and egg. There's also regeneration or regrowth of body parts. Sadly, we don't do this, but starfish could do that, or a worm called planaria. That is a flatworm that you can see over here. It's actually possible if you cut a planaria in half, they can actually regrow their entire body and become two worms. It's pretty cool. But more things than just starfish and planarias can do this. Lizards can regrow their tails, newts, lobsters, crawfish, a bunch of others. So the cells in our body, they go through a cell cycle. And that isn't just mitosis. There's a whole lot more to the cell cycle besides division, because division is just one part in what a cell does day to day. And each cell is only going to divide one time effectively, because one cell is going to replicate itself and divide into two. And now you have two completely new cells by the end of that. So mitosis effectively represents one end to a cell's life before that cell divides and becomes two new ones. So this is a graphic that you're going to see a lot, or some sort of graphic like this. We have the main part of a cell's life, and that's interphase. So that's this, imagining that a cell's life cycle was this entire circle. Interphase would account for like more than 3/4 of a cell's life. During interphase, you have the G1 phase, which is the period of cell growth before you get any sort of DNA replication. So right after a cell is formed, G1 is occurring. Then you have the S phase, and during that period, DNA is replicated. That is going to take a while, because remember, you have a lot of DNA in your cells, so that replication takes a long time. And then G2 here, and that's the period after DNA is replicated, where the cell itself is preparing for division. Because remember, there's not just DNA in your cells, there's ribosomes, there's endoplasmic reticulum, there's Golgi complexes, there's a bunch of stuff in there that needs to prepare for division as well. And then after the G2 phase, then you actually have the active dividing, or mitosis, which is split into prophase, metaphase, anaphase, telophase, and then finally cytokinesis. So mitosis itself is the division of the nucleus, not the entire cell. And I'll explain why in a little bit, but the nucleus is arguably the most important part of cell division, because that's all the information that that cell needs to function properly, that has to be divided correctly. So prophase is the first part of mitosis, and during that phase, your DNA is coiling into chromosomes. So it's that chromatin is coiling and coiling and coiling around histones to form itself into chromosomes. The nuclear membrane is disappearing. So the nuclear membrane, the wall of the nucleus effectively, that goes away. Centrosomes are these organelles that don't really have a purpose other than during cell division. They produce spindle fibers that are going to pull those chromosomes to opposite ends of the cell. Those are going to go ahead and move to opposite poles to prepare for that. And then spindle fibers are forming because of those centrosomes. Then next you have metaphase. I always remember metaphase because it's middle. It starts with the same letter. Your chromosomes are going to line up on the middle of the cell. So you can see that happening here and here in these two cells. They line up beautifully along the middle, and those spindle fibers are going to attach themselves to the centromere of each one of those sister chromatids there. Then during anaphase, I remember anaphase because those chromosomes are going to get pulled away from one another, so anaphase away. The chromatids of each chromosome are separating at the centromere, and those spindle fibers are pulling them to opposite ends. The centrosomes that are on each opposite ends of the cell are turning and pulling and cranking those spindle fibers to pull those chromosomes apart. And then during telophase, the spindle fibers are going to start to disappear. So during telophase, your chromosomes are on opposite ends of the cell. They're going to thin back out into chromatin, into regular globby DNA, and that nuclear membrane is going to reform around that glob of DNA. So now you have a nucleus again. So here you can see it. This here, before the nuclear membrane is disappearing, this is interphase, when there's DNA replication going on and all that normal cell function. Then after that, you get prophase, metaphase, anaphase, and finally telophase. So here's a nice graphic, and you can see it in real pictures of real cells that have been dyed, and over here as well. Whoops. Cytokinesis happens after telophase. So after the nucleus starts to reform, after mitosis is technically over, the rest of the cell still has to divide. So during that, what we call a cleavage or a cleft forms between the two new cells that have not divided yet. In plants, it's going to be a little different. In plants, you get this little cell plate like you see over here that's forming to split the cells in two. But cytokinesis is important because you have to split up all the rest of the organelles, and that's how it physically separates the two cells from one another. But remember that cytokinesis is completely separate from mitosis itself, because mitosis is the division of the nucleus and cytokinesis is the division of everything else. So here you can see the different phases happening, and I'll just kind of quickly go through this. You could use this as a study guide later. But you can see the centrosomes, you can see the chromosomes starting to form here. You can see the spindle fibers showing up, then they're forming over in prophase, but you can see them connecting there. You can see anaphase, those spindle fibers are pulling the chromosomes away, and telophase, and then finally cytokinesis there. Something else to talk about beyond just mitosis, what about when it goes wrong? What about when something messed up happens? And I have to talk about proto-oncogenes for you to really understand that. So proto-oncogenes are genes that regulate cell growth, and they code for the production of regulatory proteins that essentially ensure that everything is going right. So we have genes to regulate mitosis, to regulate cell growth, to regulate cell development, etc. And so if those genes go wrong, then they become oncogenes, and then you get cancer. Proto-oncogenes, you need these because they're regulators. But if the regulators stop doing their job, then bad things happen. So normal proto-oncogenes, they can become oncogenes. It doesn't necessarily mean that they will. We all have proto-oncogenes in us. They're necessary. Some of us may also have oncogenes in us. Those are proto-oncogenes that have been mutated. But that doesn't necessarily mean just because you have an oncogene, you're going to get cancer. That's not what I'm telling you. You need a series of oncogenes to definitively get cancer. So mutations happen all the time for many reasons as well. So like it said before, and let me go back for a second, it says it only requires one mutation to turn a proto-oncogene into an oncogene. But again, that doesn't necessarily mean you'll get cancer. Oncogenes can cause uncontrolled cell division or cancer, but they don't always cause uncontrolled cell division and cancer. If you have a series of oncogenes that are all kind of working together, then you end up with cancer. Just having one does not necessarily mean that you will have cancer. Why though? Because you have these things called tumor suppressor genes that are, they're the brakes. They're the, whoa, hold on, slow your roll genes. So those code for proteins that prevent cell division from happening too often. So essentially, let's say that there's a mutation in a proto-oncogene, it turns it into an oncogene. That oncogene is stimulating cells to overdevelop or over divide or whatever. You also have tumor suppressor genes that go, whoa there, slow down, why don't you stop right now before it turns into cancer. So oncogenes result from the activation or turning on of proto-oncogenes. They only need one mutation to occur. So examples of those, BRCA1, RB, APC, P53. You could look these up if you want to see what specifically positioning they have within our cells or their specific jobs. But tumor suppressor genes, on the other hand, they cause cancer when they are inactivated or they're turned off. These need two mutations to inactivate that gene. So SRC, RAS, HER2, MYC, those are all examples of tumor suppressor genes. So when you have a tumor, you have the abnormal proliferation or growth of cells from uncontrolled cell division. So in this case, we see a tumor that has formed in this lady here. She got it removed. Happy times. Tumors are not always considered cancerous or malignant. Sometimes they're benign tumors. Like this, if I had to guess, was probably a benign tumor. That doesn't mean it didn't cause her any issues. It just means that that tumor did not spread to several other places in her body and form new tumors as well. So when you are a benign or a non-cancerous tumor, the cells remain within the mass and they don't really pose a threat to your life unless they're forming like in the middle of your brain or on your vital organs or somewhere dangerous like that. But they grow. And the reason that they're growing is because most tissues in our body have a thing called density dependent inhibition. So that's a result from those tumor suppressor genes and all of that. But basically, when the cells hit a wall or have reached the capacity that they're supposed to, they stop. But when you turn that off or you inactivate your tumor suppressor genes, then you get an inactivation of that density dependent inhibition and you get your tumor. When you have a malignant tumor, on the other hand, and you have a tumor that's growing, if those cells are liable to break off and travel somewhere else and then form a new tumor, that's when you have cancer. That's when you have a malignant tumor. And those are generally considered much, much more dangerous because what's more dangerous, a tumor that's in one spot or a tumor that also is in 50 other spots? So when those malignant cancer cells, they break off of the initial tumor and they travel somewhere else, that is called metastasizing, or the invasion process is the metastasis that's breaking off of the initial and moving somewhere else. You can diagnose that by a biopsy, taking a tissue sample. You can diagnose that through certain scans as well. If like PET scans, sometimes people will take a PET scan and they'll have to drink a slightly radioactive fluid that's going to bind to certain things in your body and light up. It's not dangerous to do. It's a harmless form of radioactivity, but you can see it on a computer, and so things that are not where they're supposed to be or tumors will light up on that PET scan so that doctors can see them. Once again, what causes cancer? Spontaneous mutations of genes that code for those regulatory proteins. Those mutations can be caused by certain things called carcinogens. And carcinogens is anything that can mutate DNA or can mutate a gene. So that could be a lot of things. Anything that can cause cancer. Radiation is one of them. So UV rays from the sun, that's why they tell you to wear sunscreen. If you're outside and you're allowing that radiation to penetrate your skin, you are encouraging the mutation of your own DNA. So here in this picture, we see different moles These are generally considered dangerous moles if they are asymmetrical, if they have an irregular border, if they have different colors to them, or if they're growing. That can also be dangerous. But don't look at yourself right now and then assume you have cancer just because you have a mole that fits one of these criteria. If you're concerned about any one of the moles on your body, then go get it checked out. And that's really not going to be that big of a deal because doctors like it when you're proactive with your own health. So if you're concerned about anything that may or may not be on your body, get it checked out. Just because you have an asymmetrical mole does not mean that it is cancer. It's just one of those things that doctors look for, especially in people that are getting older. If they start noticing moles popping up, they might want to check that to make sure that it's not a cancerous mole. X-rays are another thing that can cause things like this. Chemicals, tar, nicotine, that's a popular one. So you guys have heard this time and time again, smoking will cause cancer in most cases. This is a smoky lung. This is a healthy lung. This is disgusting. This is healthy. Another chemical that when breathed in can cause a lot of problems, asbestos, which is carcinogenic and found in old construction materials. But certain viruses and certain bacteria can also be carcinogens. HPV, or human papillomavirus, which causes cervical cancer, is a carcinogen. Hepatitis B or C that can cause liver cancer, those are carcinogens. And Helicobacter pylori is a bacteria that it can cause stomach cancer and a lot of other problems. It also causes ulcers, by the way, and it reminds me of a funny story. The guy that discovered that Helicobacter pylori causes ulcers, he had to test this. And so his really great idea for testing whether or not Helicobacter pylori caused stomach ulcers was to drink a vat of this bacteria and give himself ulcers to prove that it caused stomach ulcers. Is that the smartest decision on planet Earth? Absolutely not. This was a horrible choice. Don't ever do that if you wanted to test something. I just find it hilarious that he was like, you know what? The only way I can test this is to drink a vat of bacteria. Sounds good. It was a poor choice. But anyway, here's some nice images. So this is some of the different outcomes of Helicobacter pylori. So you could have asymptomatic chronic gastritis or swelling. You could have atrophic gastritis, which this does not look very pleasant. It just looks irritated and raw. You could have an ulcer, which is what this is, bleeding in your stomach or intestinal lining, or you can have cancer as a result from all of these things. So if you did develop a stomach ulcer, you probably want to treat that. Don't just continue letting that be a thing. This is a picture of cervical cancer caused by HPV. You can see that it's cancer because cervixes are not meant to look like this. They're not meant to be all bumpy and gross looking with all of these lumps and weird colors on it. Here is a computer generated image of a liver with hepatitis, and then a liver with liver cancer caused by those hepatitis virus particles. And then here's an actual picture of a liver with cancer in it caused by hepatitis. And then the last thing that I want to talk about is apoptosis, which is a term you need to be aware of. That is programmed cell death or cell suicide, essentially. So if a cell becomes damaged or it's not working properly or whatever, there is a system in our body called apoptosis that can program that cell to die. And usually this works really well for regulating the growth of tumors or uninhibited cell growth or anything like that. So if any of your cells become damaged, they then essentially commit suicide to save your life. But that doesn't always work out. And so if the genes that regulate apoptosis become damaged and those cells do not die, then after that, that's when you would get tumors and cancer and all those issues. So like I said before, I'm going to do another PowerPoint presentation to talk about meiosis. This just really talked about mitosis by and large, and So look forward to that. I'm going to post this on YouTube, and also I'm going to post a link to it on my teacher page. If you have any questions, you don't know how to deal with stem scopes or whatever, just email me. I'm happy to answer any questions as you have them. Stay safe and have a good rest of your day.